This is a video review of DeepKymoTracker, the software for tracking and segmenting T cells in microscopy cell movies. Here you can see the title page of DeepKymoTracker interface. The pipeline consists of 5 consecutive steps, and accordingly, the 5 buttons to the left are provided, which allow you to navigate to the step you need. Let's have a look at input movies first. Each frame of an input T-cell movie needs to be represented in 2 channels, the bright field channel and the green fluorescent one. The red fluorescent channel is not needed for the algorithm to run, so it can be absent. But if it is present, even in some occasional frames, DeepKymoTracker will output the results for this channel as well. In some movies, like this one, there can be 4 wells. In others, only 1 well. You can immediately see quite a pronounced wobbling of the image due to the microscope camera instability. This wobbling may negatively affect the tracking results, as in most of the cases we need not only the areas, the perimeters, and other characteristics of the cells, but also their accurate positions inside a well. That is why, to eliminate this wobbling, it's necessary to prepare the movie for tracking and segmentation first, by cropping out the well of interest. That is what step 2 of the pipeline is designed for. It's called cut one well. The goal of DeepKymoTracker is not only to segment and track the cells, but also to build the lineage tree. Here's how the output movie looks like. The frames of the movie to the left are the images which show only 1 well. Unfortunately, it cannot be seen here because this is a fluorescent channel. This well was cropped out from each frame, and this cropped movie was passed to the tracking and segmentation algorithm. To the right, you can see the lineage tree, which has been built dynamically. Apart from this visual output, the program also calculates a number of characteristics of each cell, such as the cell area, the perimeter, the centroid, the bounding box, the average intensity of the cell body in each channel, etc. These numerical results are recorded in an Excel file. This is an example of Excel file for each cell. Let's get back to the title page of the interface. The first 2 steps in the pipeline are the preparatory ones. They allow you to prepare the input raw cell movie for tracking. The main part of the algorithm is step 3, where the pre-trained convolutional neural networks perform automatic tracking and segmentation of T cells in an input 1 well microscopy cell movie. This step is also provided with the tools that allow the user to pause tracking and manually correct the tracking errors such as cell ID swapping, misdivisions, the emergence of new cells, and cell deaths. Step 4 is designed to help you correct the segmentation errors and can be applied only to the movie which has undergone the previous step 3. That is the movie that has already been tracked and segmented. You may notice that in this pipeline, segmentation follows tracking, not vice versa, which kind of runs contrary to the convention. The explanation is that in DeepKymoTracker, tracking is the first step and segmentation is the second. That is why the manual corrections of tracking and segmentation errors should be done in the same order. Finally, step 5 allows you to visualize some of the aspects of the tracked cells. In this video, I'm not going to cover step 1 because it's all about bringing an initial raw input movie to the required format. Here is the input movie we are going to work with. It's not even a proper cell movie, it's just a short section of 63 frames represented in all 3 channels. You can see the red fluorescent channel to the right, even though it's not needed for the algorithm to run. Start with step 2. We are now in step 2 window. Step 2 allows you to crop a well of interest out of the initial raw cell movie. The program crops out the well by means of the flood fill algorithm. Even though the flood fill is applied separately and independently in each frame, but lighting conditions for a particular movie can cause a systematic error. Therefore, to minimize this error, it makes sense to crop out the well from one frame first. then manually correct the error if necessary, and then calculate the parameters for correcting this error so that they could be applied in the remaining frames. That is why the main procedure we are about to start applying happens in these two steps. There's a black field at the bottom of the window for the user instructions and feedback from the program. Let's read these instructions. To choose raw movie, press button 1. It is here. Then navigate to your raw movie, open it, and click on any bright field image. So that's exactly what we are going to do. So this is our tutorial folder. And here is the movie we need. So we click on that movie, it opens, and we need to click on any bright field image. Okay. It is displayed in the left window. Our next task is to threshold this image. To this end, we need to click somewhere on the border of the well to measure the intensities of border pixels. And after 2 clicks, button 2 starts flashing. Let's press it. And we will see the thresholded image in the middle window. Now you can use the provided slider to improve the thresholding. Your goal is to make the border of the well of interest one solid continuous line without gaps. The next step is to try and detect the well you are interested in. To do that, just click somewhere inside the well here. You can see the flat fill algorithm is applied to that well, and a red frame appears around the well, which is going to be cropped out. As you can see, in our case, it's exactly what we need. The well of interest has been detected correctly. Actually, you can click inside other wells, and they are detected correctly as well. But let's pretend that this is our well of interest. So we finally detected our well correctly. Now we can crop it. Let's push this button, and the cropped well appears in the middle window. It's very, as I said before, it's very important to do the cropping as accurately as possible. Therefore, let's push this button, and here you get the magnified version of the cropped well. If you notice some imperfections here, you can improve things by dragging the image with the mouse. So, for example, here, I position it here. Then we close the window. And now is the time to crop the same well from all the other bright field frames of the movie. Let's push this button, and the process begins. You can watch the progress in this window. After the process is finished, we use the slider to scroll through the cropped frames. You can see that the wobbling has been significantly reduced, but still some frames maybe need improvement. For this purpose, let's push this button. And again, we get the magnified version of the cropped bright field channel. You can scroll through the frames using the slider, and once you detect the frame that needs improvement, you can use the same technique using the mouse to drag the image to the desired position. Then you can do the same thing with other frames. So you can repeat this process as many times as you wish. Once you are finished with editing, remember to close the window. And now we are back in the main window, and we are ready to crop the well from the green and the red fluorescent channels. Let's push this button, and the algorithm starts cropping the well from those channels. And you can watch the progress in the first 2 windows. Again, when everything is finished, you can scroll through the frames again to see how the whole one well movie looks like. This is the zoomed in version of the cropped movie we just created. As you can see, the wobbling is significantly reduced. Although some frames could be probably improved. But we are going to proceed to the next step of the pipeline. Okay, let's go to step 3. Step 3 is the core part of DeepKymoTracker. It is in this step where the automated tracking, segmentation, and division detection happens. First of all, let's load our movie. We need the movie with the name One Well Movie Tutorial. This is the movie we just created in the previous step. The background color of the window has changed all of a sudden. Why is that? This is actually a new window. Let's call it the preparation window. Before we can launch tracking, we need to pass a number of parameters to the tracking algorithm. And we are going to assign them manually in this window. To begin, let's push the button which says Enhance Initial Image Contrast. Enhance Image Contrast. The possibility of changing the image contrast was implemented solely for the user's convenience. The tracking algorithm doesn't need this. It will still be using the original non-enhanced images. The reason is that in some movies, the green fluorescent channel is so low contrast that the outlines of the cells are barely legible by the naked eye. And this can cause serious inconvenience as the user is supposed to monitor the progress of tracking visually in real time. So you can play with the slider to adjust the contrast, and after you are happy, you can push save. Now we need to measure the cell size. Why do we need it? The average cell size can vary from movie to movie and therefore needs to be measured for each movie separately. We start by clicking on a few cells in this frame. The red circles and their yellow squares appear around each cell. What do they mean? The diameter of the red circle is the average diameter of the cell, which will be passed to the algorithm, and you can adjust it by using this slider. So this is one of the parameters which we need to assign. The size of the yellow square is the patch size, which will be passed to the algorithm as well. This is the second parameter. To understand the meaning of the patch size, let's have a look at the pipeline of DeepKymoTracker. The tracking and segmentation in DeepKymoTracker happen in two steps. The first step is tracking. The tracking neural network calculates the centroids of these cells, and then the algorithm cuts out the patches with these centroids, and these cropped patches are then passed to the segmentation neural network. And the segmentation neural network segments these patches. So the size of the yellow square is just the patch size, which will be passed to the algorithm. Now the question is, what is the right size of the red circle and the yellow square? The main thing you need to aim for is that the patch size is big enough to incorporate the whole body of any cell in the movie. As for the red circle diameter, it's not so crucial. As you can see, one of the cells here doesn't quite fit the red circle, and this is absolutely fine. So to make sure that you adjusted the sizes correctly, you can scroll for the frame and keep clicking the cell. For example, this cell looks a bit too long. That is why we can adjust the cell diameter, and so on and so on. You can keep clicking until you're happy, and then we need to save. So now is the time for the last parameter, initial cell positions. So what you can see here is the very first frame of our movie from which the tracking will start. You need to decide how many cells you want to track. You can actually track any subset of these cells, 2 cells, 3 cells. You can also track all the 8 cells here. The tracking algorithm can tackle any number of the cells. In this video, however, I'm going to click only on one cell because the more cells the tracking algorithm has to process, the slower it becomes. And my old desktop is not powerful enough to cope with many cells. It slows down quite dramatically, which would be quite annoying. But if your machine is more modern and more powerful, and especially if it has at least one GPU, everything will be working just fine. So now I'm clicking on this cell and close this window. Now we are back in the main window and everything is ready for tracking. So we can finally push execute button. The automated tracking starts. Your task as the user is to monitor the progress happening in the middle window. And if something goes wrong, you need to pause the algorithm, make necessary corrections, and relaunch execution again. Note that you can correct only tracking errors here, such as listed here. edit ID swappings, edit misdivisions, add new cells, and remove dead cells. As for the manual segmentation correction, it can only be conducted at the next step of the pipeline, which is step 4. So, while monitoring the tracking progress in the middle window, you need to concentrate on the above-mentioned tracking errors only, ignoring segmentation imperfections completely. So you can already see some segmentation errors here, but unfortunately we can't correct them here. We leave it for the next step. So let's learn now how to correct these tracking errors. If you spotted some tracking error here, you need to pause the tracking, display results, and after that you need to navigate to the frame where this error happened. So let's practice some of these tools. Let's imagine that we want to correct misdivision. Of course, no misdivision happened here in this particular movie, but let's pretend for the learning purposes that it did happen in this current frame. Let's imagine that the magenta cell was supposed to split into these daughter cells, but the algorithm failed to detect this event. What do we need to do? First of all, we need to activate this tool, which is called edit division. And then the program needs to know two pieces of information. First, who is the mother cell? And second, where should the daughters be? What are their locations? The mother cell is now in the previous frame window, and the daughter cells are in the current frame window. So to inform the algorithm of who the mother cell is, you need to left click on the magenta cell, on the mother cell in previous frame. Then move to the middle window, to the current frame. And here we need to click on both daughter cells. We are clicking on the centroids, and that's it. Now we need to close the editing mode and relaunch the algorithm, the tracking once again. After a while, we can see the following picture. The magenta mother cell disappeared and two new cells came to existence instead, these orange and yellow daughters. The division can be clearly seen in the lineage tree as well. The magenta branch has split into two sub-branches corresponding to those yellow and orange daughter cells. Let's practice one more editing tool. Let's pause the algorithm again. Go to some frame. And let's suppose that we need this editing tool, which says add cell. Why do we need this tool? The thing is that sometimes cells which are moving in different wells, they can jump over the well. And that is why a new cell may appear in the field of view out of the nowhere all of a sudden. As the tracking neural network we utilize here hadn't been trained to detect new cells, you need to add that new cell or maybe multiple cells manually. That is why we pause the algorithm. And let's suppose that this new cell is this one. We want to add it. What do we need to do? We need to activate this editing mode, which is called add cell, and just click on that new cell, and that's it. Let's save and relaunch our algorithm once again. And now we can see that a new cell appeared in the field of view in our tracking algorithm, and also a new branch corresponding to this new cell has appeared in the lineage tree and started growing. So it's not necessary to wait until the end of the movie, actually. You can pause and interrupt it anytime. So to save our tutorial name, that's exactly what I'm going to do. I'm pausing the algorithm. Do this once again. And you can see some segmentation imperfection, but we are going to correct them in the next step. So now we are ready to proceed to the next step. First of all, we need to save the movie, even though it's not finished. So the movie has been saved, and now we can proceed to the next step of the pipeline. Now is the time to correct segmentation errors. There are two segmentation techniques available, as you can see from the screen, fast technique and the slow technique. The fast mode, this is a very fast and nice technique because it doesn't require a lot of effort from the user. all you need to do is just keep clicking on a cell until you obtain the optimal segmentation. As for the slow technique, slow mode, it's just a traditional hand drawing with the mouse. You can also zoom in and out here, and you have the pen tool here. These are traditional helper tools needed for manual segmentation correction. You can also switch from one channel to another. Okay, let's start by loading our movie. We need to find the movie with the name Tracked Movie Tutorial. This is a movie we just created in the previous step. So our movie is loaded now, and we can use the slider to scroll through the frames. As you can see, there are two windows here. Each frame is given in two channels, the green fluorescent to the right and the bright field channel to the left. The active window, the window where you're going to make correction, is always the right window. So the fluorescent channel is active now. If you prefer to work with the bright field channel, you can use these buttons. You can actually switch from one channel to another anytime you like, at any stage of your correction. So you start. So we immediately see the segmentation error. How do we need to correct this cell? First of all, you need to right click on that cell, and then it's a good idea to use zoom tool immediately. And now, as you can see, fast mode is activated. That is done because it makes sense to start with the fast mode, and only if you don't manage to achieve good segmentation. Only after that you will have to resort to the slow mode. So there is a cheat sheet provided for your convenience, and cheat sheet says that to create a contour you need to left click on the cell. So let's start left clicking on the cell. And you can immediately, we need to left click at different locations of the cell. And you can see how the segmentation changes with every click. So the question is, what's going on here? Why does it look so nice? The secret is that every time you left click on the cell at different locations, the pre-trained segmentation neural network is applied to perform the segmentation of this cell. It is the same neural network which was utilized during automated tracking in step 3. The segmentation of the cell obtained in that step wasn't good enough because the tracking neural network calculated the centroid of that cell not accurately enough, and it passed that not very accurate centroid coordinates to the segmentation neural network. In other words, the segmentation error happened due to the tracking error. And what we are doing now when we keep left clicking on the cell, we are trying to correct this tracking error. The segmentation neural network takes the coordinates of your clicks as the centroid's coordinates and tries to segment that cell using these coordinates. So our clicking is essentially a trial and error method. So as you can see, we did achieve quite a good segmentation, and now we need to save by right-clicking it. So this cell is here. Let's move to some other. So we can repeat this process. So right-click on the cell, start zoom, and again, we are in the fast mode. So let's try and click on the cell. Oh, and again, we almost got a good segmentation almost immediately. So let's move to some other frames. Here we can have even 3 cells. So as you can see, this fast technique is really nice and fast. But what if you didn't manage to achieve good segmentation? Let's pretend that we are working with this cell. Okay, right click on it, start zoom, and let's start left clicking on it. Okay, it makes sense to change the channel. And maybe this is just the case when we can't achieve a good segmentation. I think that the outlines of the cell are here. So I'm trying to click, 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 and I still not able to achieve this good segmentation. What do I need to do? I need to switch to the slow mode, activate slow mode. As you can see, the red contour disappears to make room for the new contour. And now, according to the cheat sheet, to create contour, you need to draw with the left mouse. So I'm drawing with the left mouse. And here we are. Now we can save. And we are back in the big image. So that's how this correction is done. So you can do as many corrections as you wish, as you need. going from frame to frame. You can even interrupt the process and leave it for the later time. All the information about new segmentation will be recorded. Okay, so let's create final movie now in Excel files. Let's pretend that we are finished with segmentations. And now we are ready to proceed to the fifth step of our algorithm. We have finally reached the last step of DeepKymoTracker pipeline, step 5, which is called visualize results. First of all, let's load our tracked and corrected movie, which is called tracked movie tutorial. So the movie is loaded now, and the menu choose cell ID started flashing. Let's open it. Here you can see the list of our cells, and let's go for cell 11, for example. Do you remember it was a yellow cell, yellow daughter? Now we need to choose the cell property. Let's go to the area. And here they are, the visualizations for cell 11. In the top left window, you can see the tracked frames of the movie given in the bright field channel this time. Only the frames where our cell 11 was present are displayed here, and you can use the slider to scroll for them. When you move the slider, you can watch how the location of our yellow cell and its relative position with respect to the neighboring cells is changing from frame to frame. On the right, you have another window with the magnified image of the yellow cell. Again, using the slider, you can get a sense of how its shape is changing. In the middle window, we have the lineage tree of our movie, where the position of our cell in the tree is indicated by the red marker. So you can see how this red marker is traveling along the yellow branch. Finally, here in the bottom left corner, we have the diagram of the area of our cell in each frame. And the current value of our cell is highlighted in red. You can also see the other values, the values of other parameters of our cell here in the center of the screen. And the current parameter, which is displayed in the diagram currently, is highlighted in red. If you switch to the perimeter, for example, of the same cell, we have a different diagram here. This is diagram for perimeter values, of course. And the perimeter value is highlighted in red. But as for the first three windows, they do not change because this is still the same cell. Let's explore some other cell, maybe cell A. We need to choose property first. Let's go for the circularity this time. And now we have all the visualizations concerning the green cell. You can see this red marker traveling around the green branch of the yellow of the lineage tree and so on. So these are visualizations for each particular cell. Apart from this visualized information, we also have numeric information created by the DeepKymoTracker. Let's have a look at it. So here we have a list of our cell identities, and during the execution, the program creates a separate folder for each cell. Let's go for this yellow cell 11. Inside this folder, you will find lots of other folders with all sorts of visual information about this cell. But we are going to open Excel file this time. Here in the Excel file, you can see lots of numerical information about our cell. Cx and Cy are the centroids of this cell in each frame given in pixels. The next columns are the area, perimeter, and circularity of our cell. Then we have the bounded box parameter. And also we have the average intensity of our cell body with respect to each channel. And also we have very pretty familiar diagrams for the area, perimeter, and circularity.